Okay, so welcome back to this next video in which we are discussing cyclic AMP signaling. Okay, right, so we have now looked at the GS cascade, okay, and we've seen the main target for the cyclic AMP that is produced by the GS cascade, which is protein kinase A. We've also now looked at how the signal is terminated once the activating ligand that is driving the GS cascade is removed from the extracellular fluid. What I now want to look at is a really important example of where this cascade is used in physiology, okay? And that is in the response of the hepatocytes in the liver and the skeletal muscle cells and the adipocytes to adrenaline. Okay, which is an important hormone in the blood. Now, adrenaline is also called epinephrine, okay, in the States. So, adrenaline or epinephrine, whichever name you prefer, we're talking about that. Okay, and basically, adrenaline and epinephrine is released into the blood by the adrenal medulla, and we'll go through this in a bit more detail in a moment, okay? And it has effects on the hepatocytes of the liver, the skeletal muscle cells, and the adipocytes, okay? And basically, what we're going to use this as an example to highlight is a very important principle, which is the principle that the effects of this same cascade occurring in different cells can be very different. Okay, so in hepatocytes and in skeletal muscle cells, the adrenaline is going to promote glycogenolysis, the breakdown of glycogen, whereas in the fat cells, the adipocytes, it's going to promote the breakdown of lipid molecules, lipolysis. Okay, and we want to look at the different pathways in these three different cell types, okay, to highlight the fact that this same pathway can cause very different effects in different cell types. Okay, right, so let's start with the big picture. Where does adrenaline or epinephrine come from? Okay, so basically it's released by the adrenal medulla in response to sudden stress. Okay, and I've missed out a D there. Sudden stress. Okay, so for instance, if you're about to be attacked by something, if you just notice a bear coming up behind you or something along those lines, a life or death situation, okay, that counts as a sudden stress. Then what's going to happen is the adrenal medulla of your adrenal glands is going to start releasing adrenaline or epinephrine into the blood. So let me just draw a little picture of the adrenal gland. Okay, now you have two adrenal glands, one on top each of your kidneys. Okay, and the adrenal gland can be divided up into two main portions. Okay, so there is the adrenal cortex, which is the outer portion, and then there is the adrenal medulla, which is the inner portion. Okay, so the portion right at the center of the adrenal gland is called the adrenal medulla, and it's this portion, the adrenal medulla, which is capable of producing adrenaline and releasing it into the blood. Okay, so I'll colour in the adrenal medulla in red here. Okay, right, so the adrenal medulla cells, which are known as adrenal chromaffin cells, okay, so the special type of cell that produces adrenaline in the adrenal medulla is called adrenal chromaffin cells. Okay, these produce adrenaline and secrete it into the blood in response to a sudden stress. Okay, now what does the adrenaline do? Well, it does a huge number of different things. For instance, it raises your heart rate, okay, and also the force of contraction of your heart to increase your cardiac output. But we're going to be particularly interested in the effect that it has on the hepatocytes, the skeletal muscle cells, and the adipocytes. Okay, so what does the adrenaline do to each of these cell types? Well, let's give them motivation first. So basically, if you're in this life or death situation, you're going to either need to be ready to fight the scary stimulus, okay, or you're going to need to be able to run from it. So you're in basically a fight or flight situation. Now, both fighting or running is going to require your skeletal muscle to be working very hard, okay, because to move you need your skeletal muscle to contract. So your skeletal muscle cells are going to be requiring lots of energy to do that, so they're going to be requiring lots of fuel, lots of glucose and fatty acids. Okay, so basically the responses that we're going to look at in the adipocytes, the hepatocytes, and the skeletal muscle cells, they're all going to be about getting glucose and fatty acids to the skeletal muscle cells, which they can use as fuel, which we're going to need if we're going to fight or flight, basically. Okay, so how is it going to do this? Well, in the hepatocytes, the cells of the liver, the adrenaline is going to activate the breakdown of stored glycogen. Okay, and the breakdown of stored glycogen is called glycogenolysis. Okay, so breakdown of glycogen. And basically, the glucose that you get from glycogenolysis in the hepatocytes is then going to be released into the blood to be delivered to the skeletal muscle cells. Okay, so the effect of adrenaline on the hepatocytes is to increase the breakdown of glycogen so that you can produce glucose released into the blood. And we will go through the pathways of this in more detail in just a moment. Okay, in the skeletal muscle cells now, when adrenaline acts on the skeletal muscle cells, again, it is going to promote glycogenolysis. Okay, skeletal muscle cells also have stores of glycogen within them. Okay, so they have glucose polymerized into glycogen. The effect of adrenaline on the skeletal muscle cells is going to be to promote glycogenolysis, to promote this breakdown of glycogen. However, they are not going to be returning it back into glucose to secrete into the blood. They are just going to be using the glucose that they get from glycogenolysis in the respiratory action. So they're just going to basically respire the glucose they can get and use that for energy because they're now going to have to contract to fight or flight. Okay. In addition, the effect of adrenaline on the adipocytes, the fat cells, is to get the fat cells to break down the triacylglycerols that are stored in them and release free fatty acids into the bloodstream. And this is a process known as lipolysis, the breakdown of lipid molecules. Okay, and again, the free fatty acid molecules which are put into the bloodstream, those will go to the skeletal muscle cells and they can be respired. Well, it's not respiration, but they can be beta-oxidized to produce energy, to produce NAD that can then go through the respiratory chain. Okay, right. What we're now going to discuss then is we're going to discuss these effects in more detail, okay? So we're going to start off with a more in detailed discussion of glycogenesis and glycogenolysis so that we can understand how adrenaline working on beta-2 adrenergic receptors on the surface of the hepatocytes and the skeletal muscle cells can trigger net breakdown of glycogen to glucose. And in the case of the hepatocytes, the release of that glucose into the blood, and in the case of the skeletal muscle cells, the use of that glucose for the respiratory purposes. Okay, right. So let's now have a look at glycogen in more detail. So I'm going to start just by making sure that everyone knows what glycogen actually is. And then we'll discuss the synthesis of glycogen and the breakdown of glycogen. And then we'll discuss what the effect of beta-2 adrenergic stimulation on hepatocytes and skeletal muscle cells is going to be, and how protein kinase A is going to mediate those effects. Okay, so firstly let me just show you my cartoon for glucose. Okay, so I'm going to show glucose just as a hexagon like so, with the 6th carbon coming off up here. Okay, so remember, glucose is a hexagonal molecule where there's an oxygen atom up here and the other 5 atoms in this ring are carbon atoms. And then you also have a 6th carbon which comes off up here. Okay, so this is going to represent glucose in my pictures. And I'll colour it in blue here. So this is a glucose molecule. Okay, now, what then is glycogen? Well, basically, glycogen is a polymer of glucose molecules, loads of glucose molecules attached to one another. Okay, so if I just show this, what you have is, here's one glucose molecule, and basically you can attach this carbon here, the first carbon of the glucose molecule, because remember the oxygen's here, and you count around 1, 2, 3, 4, 5, 6. You're going to attach this to the 4th carbon of another glucose molecule. Okay, so here is our other glucose molecule here, and this is the 4th carbon of this second glucose molecule, and you can continue this on like so. Okay, here's another glucose molecule, and it can go on and on and on. Okay, and this polymer of loads and loads of glucose molecules, this is what is meant by glycogen. Okay, and both hepatocytes, liver cells, and skeletal muscle cells have huge, great polymers of glucose stored within them, basically. Okay, right. So, firstly, let's look at the process by which you can actually build glycogen, which is a process called glycogenesis. And then we'll look at the process by which you can break down glycogen, which is called glycogenolysis. And you might be asking, well, why are we bothering to look at glycogenesis? And it's because if you want the skeletal muscle cell or the hepatocyte to have a net breakdown of glycogen, okay, then you need to activate the breakdown process. You need to activate glycogenolysis, but you have to also stop the synthesis process. You have to stop glycogenesis. Otherwise, as soon as you break down glycogen into glucose monomers, glycogenesis will just reassemble it back again. Okay, and you'll have what's called a futile cycle. Okay, and that's why we have to look at glycogenesis as well as glycogenolysis, because actually when we stimulate hepatocytes and skeletal muscle cells with adrenaline, it not only activates the glycogenolysis process but also inactivates the glycogenesis pathway, and that's how it has a net glycogenolytic effect. Okay, right, so let's now discuss glycogenolysis then. Okay, so glycogenesis involves adding an additional glucose molecule onto a glycogen molecule, onto the free end of a glycogen molecule, okay? And basically you can't just take a free glucose molecule and stick it on here. The glucose molecule needs to be attached to another molecule in order for this to work, okay? And I'll show you this other molecule now. So basically you need a molecule of UDP, uridine diphosphate, attached to the glucose molecule in order to make this work. So you can attach UDP glucose molecules onto glycogen, but not free glucose molecules. So let me show you UDP glucose. So basically, you're going to attach uridine diphosphate onto the first carbon of the glucose molecule, okay? So let me show this here. So those are the two phosphate groups of the uridine diphosphate. Okay, here is the ribose sugar, and here is the uracil organic base. Okay, so UDP stands for uridine, and uridine, like adenosine, means the uracil bound to the ribose sugar. So the organic base here, which is uracil in this case, okay, so that's a uracil organic base, bound to the ribose sugar here. Okay, that's what is meant by uridine. Okay, and then we've taken uridine and added two phosphate groups onto that molecule, here and here, an alpha phosphate and a beta phosphate. Okay, and that produces us then uridine diphosphate. And you can see that the beta phosphate of the uridine diphosphate molecule has been attached onto the first carbon of our glucose molecule here, and that's how you create UDP glucose here. Okay, right. So you can't attach free glucose molecules onto glycogen. You have to attach UDP glucose molecules onto your glycogen. So let's say we've got our glycogen molecule here and we want to grow it. What we can now do is take this molecule over to this free end here, and we can take the UDP molecule off and attach this first carbon to this fourth carbon here to extend this glycogen molecule now to a molecule with 5 glucose molecules in. So let me just show this here. So now we've got an additional glucose molecule added on to the end there, and we've now got a glycogen molecule consisting of 5 glucose molecules all polymerized together. Okay, and when you do this conversion, you will also produce as a waste product or as a byproduct a UDP molecule. Okay, so now the uridine diphosphate molecule will be free, so you'll produce this extended glycogen molecule with a UDP molecule as your byproduct. Okay, right. So this doesn't just occur spontaneously, it requires catalysis. Okay, so this reaction where you take a UDP glucose plus a glycogen and extend the glycogen by one additional glucose and produce UDP, this is catalyzed by an enzyme called glycogen synthase. Okay, let me just move this up a little bit. Okay, so a very important enzyme called glycogen synthase catalyzes this reaction in hepatocytes and skeletal muscle cells. And for short, glycogen synthase is often abbreviated down to GS. Okay, so that's present in hepatocytes and skeletal muscle cells and catalyzes the addition of the UDP glucose molecule onto the glycogen polymer. Okay, right, so that's the glycogenesis process. Let's now look at the glycogenolysis process. So now how do you break that end glucose molecule off if you want to do so? Okay, well this is done by basically adding a phosphate group onto the end glucose here. Okay, so what you're now going to do is you're going to break the bond between this glucose and this glucose here by adding a phosphate group onto that first glucose. Okay, so you're going to break that bond, and the way you're going to do this is by phosphorylating that glucose effectively and then causing the other bond to break. Okay, so here now is your new free glucose molecule, but now it's going to have a phosphate group attached onto that first carbon there. So there's that phosphate group, and now I'll colour this free glucose molecule as always in blue. Okay, right, so this product that you've produced here, this is known as glucose 1-phosphate, okay, because it's glucose just with a single phosphate group attached onto the first carbon of the glucose ring. Okay, and for short, glucose 1-phosphate is often abbreviated down to G1P. Okay, right, and of course the other product will then be the glycogen chain 1 shorter now, so we'll have gone back to the glycogen chain where we've only got 4 glucose molecules making it up. Okay, now, what enzyme then catalyzes this reaction? Okay, well this reaction is catalyzed by an enzyme called glycogen phosphorylase, okay, an enzyme capable of phosphorylating effectively glucose molecules at the end of the glycogen molecule. Okay, and for short, glycogen phosphorylase is abbreviated down to GP. Okay, so glycogen synthase then is the essential enzyme of glycogenesis. Glycogen phosphorylase then is the essential enzyme of glycogenolysis. Okay, right. So before I discuss how activation of beta-2 adrenergic receptors on the surface of hepatocytes and skeletal muscle cells can lead to the activation of glycogen phosphorylase and the inactivation of glycogen synthase, let me discuss what will happen in the hepatocytes and the skeletal muscle cells after you've activated the glycogen phosphorylase and inactivated the glycogen synthase. Okay, so once you've inactivated glycogen synthase and activated glycogen phosphorylase because of the adrenaline or epinephrine within the blood, what's now going to happen is you're going to get a net breakdown of glycogen, which will produce you lots of this stuff, glucose 1-phosphate. Okay, now what is going to happen to glucose 1-phosphate in the skeletal muscle cells and the hepatocytes? Okay, well firstly it's going to be turned into something that's more usable. Okay, it's going to be turned into glucose 6-phosphate rather than glucose 1-phosphate, which if you know your respiratory reactions, you will know glucose 6-phosphate is the first molecule in the glycolysis pathway. Okay, so when you want to break down glucose with glycolysis, okay, what you do is you first phosphorylate the glucose on the 6th carbon, basically, to produce glucose 6-phosphate. So we have just moved that phosphate group from being on the 1st carbon to now being off that 6th carbon up there. Okay, so this is now glucose 6-phosphate, and this is going to occur in both skeletal muscle cells where we've activated glycogenolysis and in the hepatocytes where we've activated glycogenolysis. Okay, now glucose 6-phosphate for short is abbreviated down to G6P, okay, like glucose 1-phosphate was abbreviated down to G1P. And this reaction, where you convert glucose 1-phosphate to glucose 6-phosphate, this is carried out by an enzyme known as phosphoglucose mutase, okay, which both the skeletal muscle cells and the hepatocytes will have. Okay, right. So now, once we've activated glycogenolysis, we can convert the product of glycogenolysis, glucose 1-phosphate, back into glucose 6-phosphate. And now what's going to happen? Well, this is where it differs between skeletal muscle cells and hepatocytes. In skeletal muscle cells, we're now just going to send the glucose 6-phosphate down the rest of glycolysis, okay, and then down the rest of respiration to produce ATP overall. Okay, but in hepatocytes, we don't want that to happen. We don't want the hepatocytes to use this new glucose that we produced. They've produced it so that it can go to the skeletal muscle cells. Okay, now, in order for the glucose to go to the skeletal muscle cells, we firstly have to turn it actually back into glucose. Okay, so we have to take that phosphate group off the 6th carbon there and return it back to pure glucose. Then we can put it into the blood and send it to the skeletal muscle cells. Okay, right, so in the liver then, what's going to happen is you're going to take that phosphate group off the glucose 6-phosphate, returning us back down just to glucose here. Okay, and that reaction is catalyzed by an enzyme called glucose 6-phosphatase, okay, which is an enzyme which the hepatocytes will have, but the skeletal muscle cells will not have. Okay, so a very important enzyme there that converts glucose 6-phosphate back to glucose by breaking off that phosphate group that's off the 6th carbon of the glucose 6-phosphate. Okay, right, and then the glucose that you've now got accumulating within your hepatocyte will then be transported out of the hepatocyte by the transporter GLUT2. Okay, so if I just draw my hepatocyte here, you'll have a transporter for the glucose, which is called the glucose transporter 2, okay, GLUT2. And that will transport the glucose out of the hepatocyte into the blood once glucose builds up within the hepatocyte cytoplasm. Okay, so here we go. Right, so that now summarizes glycogenolysis in these hepatocytes and these skeletal muscle cells. What we now want to discuss is why does activation of beta-2 adrenergic receptors on the surface of the hepatocytes and the skeletal muscle cells drive this net glycogenolysis, okay, which is favorable for a fight or flight situation. Okay, so basically on the surface of your hepatocytes and your skeletal muscle cells, you have the beta-2 adrenergic receptor. Okay, which we know is a GS-coupled, G-protein-coupled receptor. Okay, so basically when the adrenaline goes up in your blood, it is going to be stimulating the beta-2 adrenergic receptor on the surface of the skeletal muscle cells and the hepatocytes. Okay, then that's going to be activating this cascade that we have discussed at length inside your hepatocytes and your skeletal muscle cells. Okay, so you're going to be activating GS heterotrimeric G proteins. The alpha S GTP subunit will then activate adenylyl cyclase enzymes in the skeletal muscle cells and the hepatocytes. You'll then start getting the production of cyclic AMP, which will activate our protein kinase A enzymes and release the catalytic subunits of protein kinase A to go and phosphorylate serine and threonine residues within target proteins. Okay, the question is now, how are these catalytic subunits of protein kinase A going to activate glycogenolysis and inactivate glycogenesis to promote net glycogenolysis, okay, in the skeletal muscle cells and the hepatocytes, which we have seen is beneficial for this fight or flight situation. Okay, right, so how does the catalytic subunit of protein kinase A promote net glycogenolysis? Well, firstly, it is going to inhibit glycogen synthase, okay? So protein kinase A is capable of phosphorylating and inactivating. So when I put a stick with a blunt end like that, that means inactivating glycogen synthase, okay? So if I draw a little crude picture of glycogen synthase here, okay? this can be glycogen synthase. Protein kinase A is going to phosphorylate serine and threonine residues on the glycogen synthase enzyme. Okay, and these phosphorylations are going to inhibit that enzyme. It's going to stop it working. Okay, so that you shut down glycogenesis. Okay, so you're no longer making more glycogen from UDP glucose. Right. Meanwhile, the protein kinase A is going to start activating glycogenolysis by not directly activating glycogen phosphorylase, but by activating something else that will then activate glycogen phosphorylase. Okay, so this is a little bit complicated. So if we draw a crude picture here for glycogen phosphorylase, so this is glycogen phosphorylase, we want to activate glycogen phosphorylase to promote the breakdown of the glycogen, to promote glycogenolysis. Okay? Basically, we don't phosphorylate glycogen phosphorylase directly by protein kinase A. Instead, we phosphorylate another enzyme, which is also serine threonine kinase like protein kinase A. Okay, and I'll have this enzyme in turquoise here. Now, this enzyme is the enzyme which will phosphorylate glycogen phosphorylase once it is active. Okay, and this enzyme is known as glycogen phosphorylase kinase, or GPK for short. So I'll write its full name down here. So this is glycogen phosphorylase kinase. Okay, and some people will just call it phosphorylase kinase rather than glycogen phosphorylase kinase, but I'll give it its full name. Okay, right. So this is glycogen phosphorylase kinase, and it's a serine threonine kinase. Like protein kinase A, it can phosphorylate serine and threonine residues within proteins. Okay, so now here comes our catalytic subunit of protein kinase A. Okay, so this is our catalytic subunit, and I'll just colour this in purple here. Okay, and what it's going to do is it's going to phosphorylate and activate the glycogen phosphorylase kinase. Okay, so once you've activated protein kinase A, it will phosphorylate glycogen phosphorylase kinase on serine and threonine residues, and this will produce an activation of glycogen phosphorylase kinase. Okay, and now it's activated, and it's now going to phosphorylate serine and threonine residues on the glycogen phosphorylase enzyme, and then that will activate glycogen phosphorylase, and now glycogen phosphorylase is going to start breaking down lots of glycogen into glucose 1-phosphate, which can then be converted into glucose 6-phosphate by our phosphoglucose mutase enzyme. Okay, and that's how you promote glycogenolysis within skeletal muscle cells and hepatocytes. And again, I'll state it again, in skeletal muscle cells, the glucose 6-phosphate which you generate from the glycogenolysis will just be used for glycolysis to generate energy, and that is needed because we're in this fight or flight situation. Okay, and in hepatocytes, that glucose 6-phosphate that you have produced will be converted back to glucose by this enzyme glucose 6-phosphatase, and then the glucose will be secreted into the blood so that it can go to the skeletal muscle cells and be used by those skeletal muscle cells for respiration. Okay, right, so that's the effect of adrenaline on skeletal muscle cells and hepatocytes that makes use of this GS cascade. Okay, what I now want to look at is the effect of adrenaline on a very different cell type, which is the adipocytes. Okay, and in the adipocytes, it's going to promote the breakdown of triacylglycerols to free fatty acids and glycerol, which can be released into the blood and used as an energy source by the skeletal muscle cells. Okay, but we'll look at that pathway in the next video.